Alright guys, so this video is going to cover your last couple of diagrams. So far we've done a diagram of DNA replication, and then we're going to have two for protein synthesis. We have a transcription diagram, and then we have a translation diagram. Now there was an activity that we were supposed to do for protein synthesis. It was kind of similar to the DNA replication one, but in terms of the printouts and everything, I couldn't quite get it all figured out. So that's why we did the activity that you guys did yesterday with the marshmallows and the pipe cleaners. And plus, I think that's a little bit more fun than the paper models. So you kind of got to see the process of protein synthesis yesterday. So you started off with transcription, where you took the little DNA sequence and you transcribed it into mRNA. Now, instead of looking up amino acids, you did the marshmallow colors, but essentially you had the code, okay? And then you had to take that strip and then you had to kind of run it through the ribosome. And hopefully you got to see how the tRNAs kind of shift from the A site to the P site to the E site. And then you got to build your polypeptide, which was your little marshmallow chain. So we kind of did a similar activity, but these diagrams don't necessarily match exactly what we did. And so I just want to make sure that you have exactly what you need to fill out the diagrams. I'm also going to introduce a little bit about gene regulation, which are the notes that you're going to be doing tomorrow, because in order to kind of fully understand, especially this picture for transcription, there's some gene regulation information that I'm going to share with you. And you'll hear it all again in the notes, but there's a couple of things that we're going to label that are related to gene regulation. I also wanted to go over one other thing before we start the diagrams, and that is another bonus opportunity. I was actually at the school on Tuesday, and I kind of shared this with the 7th period class, but I wanted to share it with everyone and make sure that we all are on the same page. So you have another bonus opportunity for the 9 weeks. Normally you get one, but you're going to have another one, and that's going to come from your TC surveys. So in your TC email, everybody who is a student has a TC email, and you may have already seen these if you're in other dual credit classes. But you should have gotten an email about TC surveys, okay? Now, you are in 2 biology classes. You are in 1306 and 1106, so you will have to do 2 surveys. But I will give you 5 points for each survey that you complete. This is going to be a test bonus. I know some of you may have really high test averages. We can't really take you over 100, but if that's not you, then you have another opportunity to get 10 points. If you do have a really high test average, it can maybe go on a daily grade, but it won't really have a huge effect, to be honest. So what you can do is you can go in, you complete the surveys. Now, if you complete the surveys by the time they close, then I can go in and I can see who has completed them and who has not. Now, I do want to remind you that the TC surveys are about me, okay? I am the professor. I am the one who you are answering questions about, okay? So as you go through and read, all I ask is for your honesty. It is anonymous. I do read the feedback. So I know this is a little bit different of a situation, having me at Texarkana College and Miss Petty in the classroom. But as you're answering the survey questions, it's about the TC professor, which is me. So if you would just go in and do those. If you have any issues, contact the TC help desk. They're usually very helpful if you've forgotten password or something like that. But all you have to do is click on them. I'd say probably within 5 minutes you can do a survey, so 10 minutes tops, and you can earn 10 bonus points. Now, the deadline for the surveys is the 13th, because TC finishes earlier than Pleasant Grove does, and so you will need to have them done within the next week, okay? So be sure that you have those complete. Once again, it'll tell you to screenshot and email your professor as long as you complete it by the 13th. And you don't wait until the very last minute. I'm going to be checking to see who has completed them. So on the 13th, do not wait until the last minute. I'm going to say have them done by the 12th, and that way I can easily go in and see who has completed them or who has not. If for some reason you complete them on the 13th, you may want to snap that picture. You may want to email it to Miss Petty or me, either way, and that way we can give you your credit for your bonus. So once again, 2 surveys for your biology classes, and then you can earn 10 points on a test, okay? So the very last thing that you're going to be doing grade-wise for this semester is going to be this lab quiz, okay? It is just going to be a lab quiz. It's just going to be a daily grade. Remember, we're not having a test over protein synthesis and DNA, but you will see questions on your semester exam about that material, and you can kind of refer to your review to kind of see where you need to focus. This is a very important concept, but I just didn't want to test you to death, okay? But we do need to do the lab quiz, and essentially all it's going to be are the 3 diagrams. You will have one word bank for all 3, and it should be relatively simple as long as you study. Now, kind of like with the DNA replication, if you understand the process of transcription, and if you understand the process of translation, then it's going to be a lot easier to fill out these diagrams, okay? All right, so here we go. This originally came from an animation that I was unable to find. I tried to find it, I couldn't find it. You watched some videos about protein synthesis, and honestly, that's a really good video, the one with the British lady speaking. It's a really good video and shows very well how protein synthesis occurs. So I didn't have this animation, and that's why we're just going to kind of fill it out in this way, okay? So if you have a little bit of knowledge about transcription, then this will be a lot easier. All right, so the first thing that we're going to fill out is this arrow right up here, okay? You have a box, and this is the direction of transcription. So essentially the mRNA is going to be built going in that direction, okay? And just like we saw with DNA replication, that mRNA is built from 5 to 3. On this particular diagram, you don't have to worry about the 5s and the 3s, okay? We're gonna see a lot of them on the next diagram, but this one, they're already filled in. You don't have to worry about the 5 and the 3' and what's where, okay? Now, the next thing I want us to look at is this big, chunky item here, okay? You kind of have this kind of shaded, big, oval type structure. This is our enzyme, okay? This is our enzyme that is responsible for transcription, okay? And that enzyme is going to be RNA polymerase. Now, the RNA polymerase will bind to the DNA, okay? It will build the new mRNA strand, and then it will stop when it gets to the end of the gene. And so part of your gene regulation are these regulatory sequences. Now, we talked about them a little bit whenever we discussed transcription. We said that the RNA polymerase would bind to a promoter. That was kind of the sequence that signified the start of the gene. And then it would stop when it reached a terminator, okay? So that's kind of how transcription happens. Remember, we're only copying a gene. We're not copying the entire DNA sequence. So the RNA polymerase starts by binding to a promoter, and then it knows when to stop when it reaches a terminator sequence. So if you actually jump down here to this portion of the diagram, okay, it labels the promoter. So this little box down here is the promoter, and that's pointing to that regulatory sequence where RNA polymerase is going to begin, and then it stops when it reaches a terminator. So the whole purpose of transcription is to build our mRNA molecule that we can then take to the ribosome to translate, right? Now, I also want you to remember that what we're building is kind of the beginning mRNA, okay? Sometimes it's referred to as the primary transcript, and then sometimes we refer to it as the pre-mRNA. Remember that mRNA gets edited. It gets the 5' cap and the poly A tail. It also gets the introns cut out and the exons spliced back together. So what we're building is kind of our first draft mRNA, okay? And so 2 of our boxes are going to represent this particular concept. What we were building is the pre-mRNA. So one of your pre-mRNAs is right up here. There's a box in this general area, okay, pointing to the pre-mRNA. And then you have another box down here at the bottom, which is also going to be labeled the pre-mRNA. All right, so once again in transcription, RNA polymerase is going to bind to a promoter. It's going to move along the DNA sequence. It's going to build the mRNA strand, and then it's going to stop when we reach the terminator. Okay, so that takes care of a lot of our boxes right here. Now we have a couple of more that we need to fill out, okay? Now one of them is there's a box right over here. And what that's pointing to is our template DNA strand. Remember that we use the DNA to build the mRNA. So this little box over here is our template DNA. And it's pointing to that strand of DNA there, okay? Now the last couple of things that we need to label are in this area of the diagram. And this is where we're going to get into our gene regulation just a little bit, okay? So as with everything that involves proteins, it's always very specific. Our cells don't copy genes just because they feel like it, okay? Our cells copy genes when they need to make those proteins. There's no reason to copy if we don't need to make the protein, okay? So you have to understand that this is a highly regulated process. When it comes to DNA replication, when it comes to protein synthesis, all of these processes are highly regulated. And to be honest, not always fully understood, because if they were, we'd be able to cure diseases probably a lot better. Why do we make too much of a protein? Why do we not make enough of a protein? A lot of times it comes down to gene regulation. Now, part of that gene regulation is heavily focused in the transcription process, okay? Technically, gene regulation can occur at multiple levels. There can be protein modification, there can be changes in translation, but a lot of your initial gene regulation occurs at the transcription stage, okay? Now, one of those has to do with this promoter. That's part of your gene regulation, or these promoter sequences. Another part of it has to do with, this RNA polymerase knowing where to bind and being able to bind. If the RNA polymerase can't bind to the DNA, it can't copy the gene, okay? So one of the most important things to know about gene regulation are about these proteins called transcription factors, okay? Once again, proteins, always highly specific, okay? So in order for RNA polymerase to be able to bind to the promoter, there have to be other regulatory proteins in place, and we simply just call these transcription factors. There's all different types. They're very specific in what they do. But over here in this promoter region, the RNA polymerase is not even able to bind unless the proper transcription factors are in place. And so that is a big part of your gene regulation. And so one of the things that we're going to label, there's a big circle over here. If you look in your diagram, there's some other circles and other areas of the DNA sequence. And you have a little box that's right over here, and it's pointing to several of these circles, okay? So what these little smaller circles represent are our transcription factors. And once again, those are just proteins. They bind to the sequence, and they help determine which genes are going to get copied, okay? Because without transcription factors in place, RNA polymerase can't do its job, okay? Now, I believe there's one other box that we need to fill out, and it's kind of right around here. Now, this whole sequence is what we call our promoter. Our promoter is where that RNA polymerase binds. We've already labeled that down here. But typically what you'll find in the promoter region is what we call the TATA box, okay? It's a sequence of TATA that is just part of that regulatory sequence. We'll talk about that a little bit more in the gene regulation notes. So what you're labeling right here is the TATA box. So it's TATA and then the word box, okay? So once again, the whole purpose of transcription is to build the mRNA molecule, the pre-mRNA molecule, and what that involves is the RNA polymerase will bind to the promoter. The promoter includes the TATA box, and in order for RNA polymerase to bind, the proper transcription factors have to be in place. Once that RNA polymerase binds and opens up the DNA, it will move along the DNA sequence, and it will build the mRNA sequence with those complements A to U, T to A, C to G, G to C, and then it will stop when it reaches a terminator. And by the end of this process, we have created our pre-mRNA molecule, which will then go on and be edited, okay? So that should take care of our first diagram. Now we need to do the one on translation. All right, so this is the one that comes from the activity that we could have done. And like I said, it was a paper activity that was very similar to what we did for DNA replication. And so you have a diagram that looks like this. It's got lots of blanks on it. Okay, as I mentioned before, if you understand it, then it's actually a lot easier to fill out the diagram. Okay, so please study the processes. Even though you're not taking a test on it, I would encourage you to study the notes before you do the lab quiz, because that really is explaining what's going on in the pictures, okay? And that will help you to understand better where the different items need to go. All right, so we're going to start up at the top. You kind of have a big box that has blank of blank, okay? And so up at the top, what we are labeling is the elongation phase of translation. And I ran out of room up there, so I just kind of abbreviated. So up at the very top right, you have a couple of blanks. And what you're filling in is the elongation phase. So this is where we're actually building the polypeptide. The elongation phase of translation, okay? So this one's definitely going to look a little bit different from the last one. The last one was happening in the nucleus. We had the DNA, we had RNA polymerase, we had the mRNA that we were building. Now here we're actually building the polypeptide, so we're going to have some different components, okay? Now this may seem a little complicated, but it's actually pretty simple if you understand the process of translation. Now up here at the top, we have 3 steps, okay? So there's going to be kind of 3 main processes that are happening in the elongation phase of translation. All right, the first one is codon recognition. Codon recognition. So this is where the ribosome, the mRNA is going to attach to it, and that first codon is going to be read. Remember your AUG, that's going to signify the actual, what amino acid needs to be brought. And so each of those codons is going to be read 3 letters at a time. Now, the second part is going to be peptide bond formation. So remember that we're putting peptide bonds between those amino acids. So as those tRNAs are bringing the amino acids in, when they're side by side in the A and the P site, peptide bonds are being formed between those amino acids. So peptide bond formation. And I'm going to run out of room, so I'm just going to abbreviate it there. Peptide bond formation. Now, that is actually catalyzed by something called ribozyme. So if you look in your model, there's beside number 2, there's a long blank, and then underneath it, there's another little blank. That little blank on the bottom, I don't have room for it here, but it's for the word ribozyme, R-I-B-O-Z-Y-M-E, okay? And that's going to be kind of what's catalyzing that process. And then the third stage is going to be translocation. So you remember how the tRNAs are moving from one position to the next as the ribosome moves down the RNA molecule? That's what translocation is. So we're going to put translocation. Now remember, you will have a word bank. You just have to know where to put all of these words, right? So translocation. Now you also have kind of a really big, a big box again on number 3, and then you have a little box below. That little box below is for GT powered. Now GTP is going to be a molecule that's similar to ATP. It's going to transfer phosphates, and therefore it's going to energize. So that movement, that translocation, as those tRNAs are moving from A to P to E, that ribosome is moving along the mRNA, that process is going to involve some energy, and that's going to be powered by a molecule called GTP, okay? So that takes care of the top right-hand corner, and now we're going to focus on the picture, okay? So the first thing I want to talk about is that this big chunky thing here, kind of the big thing in the background, that is a ribosome, okay? Remember, it has a big subunit and a small subunit, and so to me it kind of looks like a big hamburger or something. But here you have the big subunit, and then you have the smaller subunit on the bottom. So that big chunky thing in the background is your ribosome, okay? We have our mRNA molecule. We have our tRNA molecules, and then we're going to have our amino acids. So just to kind of give you some context, okay? So we're going to start by labeling our sites on our ribosome, okay? So what's going to happen is you can see that we're kind of reading the mRNA this direction. And so these tRNAs are coming in. Remember, they come into the A site. Then they're going to scoot over to the P site, which has the growing polypeptide. And then they're going to exit, okay? So on your paper, your blanks are in different places, but they all are followed by the word site, okay? So over here, kind of in the hamburger, you have your A site, okay? Then up here, kind of above the diagram, this particular tRNA right here, this tRNA is in the P site. And then over here, where we can see this tRNA is exiting, we have our E site. Okay, so that takes care of our locations on our ribosome. So once again, there's the word site after each one of those. So A, P, and E. Okay, so we're moving kind of, the tRNAs are going to move from the A site to the P site to the E site. We know the P site always has the growing polypeptide. The A site is where the new tRNA is coming in, and then the E site is where that tRNA is exiting. And when it's exiting, that amino acid is no longer there. Remember, it's up here in that polypeptide chain. All right, so the whole big thing is the ribosome, okay? Now down here, we have our mRNA molecule, okay? So down here you can see our mRNA molecule, and our mRNA molecule is the one that has the codons. So this particular 3-letter sequence here, that represents our codon. Now, one thing you do have to fill out in this diagram are the 5s and the 3s, okay? And so you need to understand that that 5' end is what's going to be attaching to the ribosome, okay? So we're going to start with 5' on that end, and then 3' on this end, all right? And just like we saw with DNA, it's always going to be opposite, okay? Now, you do have a little bit of a clue, all right? So one thing I want you to see is that up here on your tRNA, okay, this particular tRNA, it's got it labeled, okay? It tells us that that is 5' and that is 3'. The amino acid is always attached to the 3' end, okay? It's the one that kind of sticks up. Now, I know I wrote that really small, but that's already written on your diagram. If you look, that 5 and 3 are already there, okay? So you have a little bit of a cheat sheet here. So if this is my mRNA, 5' to 3', then on my tRNA, which has that anticodon, it's going to be opposite, okay? So here I'm going to have 3', and there I'm going to have 5'. So the numbers that you're putting in here match the numbers that are on that tRNA molecule, and they are going to be opposite of the mRNA. Okay, so that should help you kind of fill those in. Now, over here on this tRNA, you also have to fill in the 5' and the 3', but you have this cheat sheet here, okay? This one right here is going to be the 5', and this one right here is going to be the 3'. All right, so that takes care of those blanks, okay? So I think that's all of our 5s and 3s that we have to fill out on this diagram. So if you can get mRNA 5 to 3, you have the cheat sheet on the tRNAs, okay? And as I mentioned, these big chunky items here, okay, these big kind of smaller hamburger looking structures, both of those are going to be my tRNAs. So I'm going to label both of those tRNA. And you have a box on each one of those structures that you need to fill in. All right, so we have our mRNA that's being read 5 to 3. Remember that the tRNAs are bringing in those amino acids. The codons are on the mRNA molecule, and the anticodon is on the tRNA molecule. And so you have a box where you can fill in the word anticodon. All right, so I think the only ones we have left are right up here, okay? So remember that the tRNAs, their job is to deliver the amino acids to the ribosome, okay? Those amino acids attached to that 3' end. And so these little shapes right here, both of these are amino acids. in each one of those, you're going to write amino acid and amino acid, okay? So the last thing we need to label, if I'm not mistaken, is you have a blank right up here, okay? And it's pointing to where those two amino acids are joined together. And hopefully by now we know that amino acids are joined by a peptide bond. Alright, so what we're seeing in the picture is the process of translation. So the mRNA gets read 5 to 3. It gets read in 3-letter sequences called codons, okay? Then the appropriate tRNA molecule that has the right anticodon will come in. It'll bind for a moment, okay? The tRNAs are going to start in the A site. They'll scoot over to the P site, okay? Then once you have another tRNA in the A site, you're going to have a peptide bond forming. Then one tRNA is going to exit, and they're going to continue to shift, okay? It's going to start with the start codon, and then it's going to end whenever the ribosome reaches a stop codon. And then at that point, you'll release your polypeptide, and it can go on and become a functional protein. So I'm pretty sure we hit all of the blanks on that one, okay? I'm not sure, but you do have these videos to go back to if you missed anything, all right? As I mentioned before, please study the processes, okay? Don't just try to memorize random words on a random picture, okay? If you understand what's going on, this quiz should be pretty easy for you to fill out, okay? So one more time, you have your quiz. Your quiz is going to be the 3 diagrams, okay? DNA replication, transcription, and translation. You will have one word bank for all 3, okay? And it will tell you how many times you're using each item. Obviously, you're going to have multiple 5s and multiple 3s, and so it'll tell you how many times you're using each thing. And then you may have a few bonus questions as well from the activity that you did yesterday. So I hope this is helpful. I hope you guys understand. If you have any questions, just let me know. Don't forget, you can do the TC surveys for an extra 10-point bonus. You do have to do 2 surveys, and I would like you to have them completed by December 12th, okay? But if you do that, you can earn 10 more points, and hopefully I'll be seeing you around campus soon. If you have any questions, just please let me know. Y'all have a great day.